Welcome back to this course on organic chemistry in biology and drug development. In the last 20 or so lectures, we were discussing the development, drug development processes, and first you have been told about the modern methodology to rationally design a drug and then finally develop it for the market. And then once we have that idea, then we went for rapid screening of library of compounds and how to make library. Then we went for different case studies of various diseases and what is the reason for the onset of the disease, and then accordingly once the target is known, then you can find molecules which are acting as drugs against those targets. And we have taken various types of diseases like the problems with the neurotransmission, then the anti-cancer, antibiotics, anti-ulcer, antiviral, and so on. Now, briefly during this journey, we have briefly described the one important aspect of drug discovery program, that is, once the target is identified, you write molecules and then do some in silico screening of those molecules and finally synthesize that. And before you go to the human trial, that is called the clinical trial, there is something called preclinical trial, where that drug which is the intended drug or the drug which has the potential, you have to check its pharmacokinetics and pharmacodynamics property, which are abbreviated as PK/PD studies. And today we are going to discuss what are these PK/PD and how they can be determined. And then, because once the drug crosses through this PK/PD barrier, then only it can go to the next level, that is the clinical trial. So, this is basically preclinical studies, and PK/PD is a very important aspect of that. Now, we have two terms, one is the pharmacokinetics, and the other term is pharmacodynamics. Okay. Now, these are abbreviated as PK/PD studies. Okay. If you remember, earlier I defined these two terms. PK is basically the pharmacokinetics, that means what the drug, what the body does to the drug. Okay. And pharmacodynamics is what the drug causes to the body. Okay. So, it is basically mutually exclusive. So, one is basically what the body does to the drug, that is kinetics, another is the dynamics, which is what the drug is doing when it enters the body. Okay. Now, usually what we have, usually our process of taking the drug is usually the oral route is preferred. Oral route is more preferred than the other intravenous IV route, simply because the oral route is simpler, no pain, nothing. But the problem is that if a drug is taken via the oral route, then it has to survive the enzymes that are present on its passage through the gastrointestinal tract. Okay. And where the stomach also, we know that is highly acidic. So, there are many barriers that the drug has to initially first have to pass, that is these different enzymes and also the pH sensitivity to the pH. And for this reason, usually what happens, the drug is either encapsulated in a pill, in a capsule kind of thing, or it could be used as a tablet, where various binding agents are given to the drug. So, the first test for a drug is that when it goes into the GI tract, the drug has to be released, number one. It has to be stable there, that means it has to be stable to pH, it has to be stable to whatever enzymes are there. Okay. And then it should be absorbed from the GI tract and goes into the bloodstream. Okay. Now, pharmacokinetics starts basically from the absorption stage, absorption, then distribution, that means distribution and then metabolism and excretion. Okay. These are the four things which fall under pharmacokinetics, but you see that before it enters into the, before it is absorbed, it has to withstand the acidity of the stomach and many of the enzymes that are present in the GI tract. Okay. So, that is what is called the pharmaceutical barrier. So, initial barrier is not PKPD, pharmaceutical barrier. I give you one example, like when penicillin was discovered, the first penicillin that was made was penicillin G, that is the benzyl penicillin, and penicillin being a very strange molecule, it is extremely unstable to pH, the acidic pH of the stomach. So, it could not be given through the oral route. There are many drugs which are like that, that oral route is not available for them because they are not stable under those conditions, the conditions that are prevailing in the GI tract, and penicillin is a classic example. So, in those days penicillin was given intravenously, but now later on a lot of other penicillins have been made which are stable to those conditions. Now, one thing is that the pH of the pH in the body, it varies from different parts, that is true, but mainly it is maintained between 6 to around 6 to 8. Like in the blood, it is about 7.2 or 7.3, 7.4, something like that, slightly alkaline. Okay. And there are other areas where it could be mildly acidic. So, usually you aim for a drug for absorption, you aim for a molecule which is which has got a pKa between 6 to 8. I will tell you why. Because once the drug, I find that the drug has passed the pharmaceutical barrier, the pharmaceutical screening test, that means it is stable under the that condition in the GI tract. Then the question is whether it will be absorbed or not. Now, for absorption what you need is that if it is too polar, that then what will happen? It will be more soluble in water, and then it will have a difficulty to cross the cell membrane, which is hydrophobic in nature. On the other hand, if it is too non-polar, too hydrophobic, then what will happen? It will be dissolved as droplets in fats. In fat, fat has the ability to dissolve, to put it into the kind of a micelle, that it can micelize the fatty compound, means the hydrophobic drug inside. So, there will be no question of distribution, absorption through the membrane. So, it should not be very lipophilic, on the other hand it should not be highly polar. So, what you have to have, that if you have a molecule M, then it will be convenient that if it exists in equilibrium between a polar form and a non-polar form. So, that means when it is required that the drug has to have some polar character to cross something, some membrane, some other organ, then if it is required, then MH plus will be the species which will cross that barrier, whatever barrier. It could be some organ barrier, the GI tract barrier, because it has to be absorbed there. If it is required that ionic species is required, then it will cross through and the whole equilibrium will be shifted to this direction. On the other hand, if it is required that that the barrier that is present to this system, to this drug is hydrophobic, then this molecule, the non-polar molecule will pass through that non-polar membrane, and then what will happen, that MH plus will move on to the left side. So, basically this is a good option, that you should have a molecule which has got some ionic character in equilibrium with ionic as well as neutral form. That is what I am saying. Now, what kind of molecule will exist in these two forms? It is usually the nitrogen containing molecules, because the nitrogen containing molecules have a basicity between say 6 to 9, I will say. Okay. Look at histidine and look at the extreme case like arginine, but the normal the nitrogen, the amines, if they are not guanidines, then they have usually the pK range is 6 to 8. So, in 6 to 8 what will happen, both the species will be present, and 6 to 8 is the pH that exists in the different locations of the human body. So, you will see, if you now look at the number of drugs and then screen them structurally, you will see that most of the drugs contain nitrogen, and that is the main reason that it can actually distribute itself into an ionic and the and into the non-polar form. Okay. So, now what happens, that if you take the drug, if you suppose this is your GI tract, this is the stomach, and then you have the large intestine, the small intestine, all this, okay. And the drug has to be absorbed from it could be from the esophagus, it could be from the mouth also. Many drugs are actually absorbed through the mouth, that is the saliva that is present in the mouth. So, the whole drug absorption process is basically the entire GI tract. It starts from mouth and then ends into the small intestine. Okay. And so, once it is absorbed, then it goes into the bloodstream, and after it goes to the bloodstream, then it has to, there is a checking point, because anything which is foreign to the body wants to reject it, whether it is an organ or whether it is a molecule. So, the body sees that some molecule has entered which is a foreign molecule. So, the drug, then the body wants to do something, basically the body wants to expel it from the body. Okay. So, if it is that is what is called metabolism. If the drug is really highly water soluble, highly polar, then what happens? The body does not have to do much, because it is already soluble in water. So, it can be excreted through the kidney very easily. But if the drug is not very polar, usually that what happens, the drugs are usually not very polar. As I said, there has to be a balance between the polarity and the non-polarity. And then what happens, the drug has to be made water soluble before the body can excrete it, and that is what is called metabolism. So, one is absorption, that is the GI tract, the drug has to be absorbed, and for that the drug has to have some balance between the hydrophobicity and the lipophilicity. Then there is distribution. Distribution is basically, it now comes to the blood stream, and wherever the blood is distributed, the molecules go there. Now, sometimes there are also some molecules which are present in the blood that can also may not allow the drug to reach the target, because ultimate aim is that it is absorbed, then it is distributed, but it should reach the target, that is important. Before reaching the target, there are some proteins which are called albumins. They are very non-specific. Many molecules can bind to the, they have the ability to bind, or they or provide surface where the drug molecules can bind. So, if the serum, this is called human serum albumin. So, human serum albumin sometimes absorbs the drug, and then the drug cannot reach the target. So, distribution is also important, whether it is really evenly distributed and then the drug has to reach the target. Now, in case where there is a problem that albumin is absorbing the drug that you are interested in, okay, drug to be distributed. So, what you can do in those cases? In those cases, what is usually done, take another molecule which has got more affinity for the human serum albumin. So, if you do that, take both the molecules together, then your drug will not be preferentially absorbed, because the other molecule has more absorption potential. So, sometimes this combination of drugs is there, that is just to stop the actual drug from binding to the serum albumin. Okay. So, that is distribution, then comes the metabolism. So, that is the bigger picture, metabolism. I said why drugs have to be metabolized, because the body treats that as a foreign molecule, and because most of them are not highly soluble in water. So, they have to be made soluble in water. Now, there are two ways of the body converting the molecule, the drug, into a water soluble fragment. One is put hydroxyl groups in the molecule, because you know hydroxyl groups enhances the polarity. But the question is why hydroxyl, why not amine? Amines are also water soluble, but the problem is where is the we have the oxygen source and we have the enzyme system to utilize the oxygen and oxidize different molecules. Okay. So, the drugs, one way that the drug has to be converted, put lot of wedge groups into the molecule, that is one way to make it polar. The other way, that is called phase 1 metabolism, and another metabolism is there where the drug is conjugated to a very polar molecule, highly polar molecule. So, there are two ways, one is the drug can be converted into wedge, or the drug can be conjugated to a polar molecule. And once this is done, then they can be excreted mainly through kidney, because now they have water soluble, unless the molecular weight is very high, then there is a problem, kidney cannot filter that. Okay. Now, we will discuss both these things. What is written here? Okay, I have already told what is pharmacokinetics, what is pharmacodynamics. So, pharmacokinetics has 4 steps: absorption, distribution, metabolism, excretion. And remember, when we talk about this first pharmaceutical test, that where it survives the acidic conditions of the stomach, that is only for orally active drugs, not for the drugs which are taken intravenously. And so, there is a way to bypass that pharmaceutical problem, because you can directly inject to the blood. Orally taken drugs must be sufficiently polar, I already highlighted that. So, that in the GI tract and to dissolve in the GI tract and into the blood supply, but sufficiently fatty, that means sufficiently lipophilic to pass through the cell membranes. Okay. So, that is the points that I have made. Now, I said that there are two ways of doing the metabolic changes in the drug. Okay. That is actually basically absorption, how the drugs are absorbed and then distributed. So, you must follow the I told you about the Lipinski's rule of 5. So, that it should be usually the drugs are less than 500, 5 hydrogen bond donors, 10 hydrogen bond acceptors, those are the upper limit, and then log p value should be less than, p is the partition coefficient between octanol and water. Okay. Log p value less than 5. So, for those it has been found that they have a very good absorption as well as solubility in the blood. Okay. And something also have added, no more than 7 rotatable bonds. Now, these are not sacrosanct, these are not written in Bible. This is basically a first screening test preliminary screening test, so that pharmaceutical companies do not have to make lot of molecules to screen out a lot of molecules to synthesize and then test and finally find that those molecules are useless. So, that is a wastage. So, the industries follow the Lipinski's rule. Now, let us see the drugs, the metabolism. We are going to discuss the metabolism, because that is where some organic chemistry is involved. I said there are two types, phase 1, phase 2. Phase 1 is put hydroxyl groups and or other polar groups, I will show you, and the second one is the conjugation with polar molecules. Okay. Now, the hydroxylation is done by, okay, so let us see, drugs, foreign substances excreted through kidneys, if it is polar, non-polar, conversion to more polar molecules, and that is what is called metabolism. But remember one thing, the drug is given for the purpose of treatment of a disease, but if it is metabolized very rapidly, that means your drug is going out from the system very quickly. So, that is also a very important issue. If metabolism is quicker than reaching the target, then the drug will be useless. So, you have to increase the dose, but then you have to again be careful that whether you are crossing the therapeutic window, all these issues are there. So, there is another big topic which is called bioavailability. That means, how long the drug exists in the bloodstream. Usually what happens, that if you are prescribed penicillin molecule, you take one in the morning and one in the means there is a gap of about 12 hours, because it has been found that the drug is sufficiently made available to the target for about 12 hours. So, you take the next dose again. So, that bioavailability, some drugs have very good bioavailability, that you take the pill only once, like the hypertensive, antihypertensive drugs, diabetes drugs, because they are more stable metabolically. So, to have good bioavailability, it has to have good metabolic stability. However, it should also flush out, because many of these drugs are not very healthy for the kidney. They affect the kidney. So, they has to, of course, they should be flushed out also. They should not remain in the system, that the disease is gone, but the molecules remain there. That also should not happen. The molecules should also have to be taken care of, so that they go out of the body. So, what happens now, that we are talking about this conversion there. Now, the hydroxylation is done once the drug enters into the bloodstream. The first organ that it crosses, where a lot of these metabolic activities take place, is liver, because liver is our storehouse of many metabolic enzymes. Okay. And one of the enzyme is what is called cytochrome P450. P450 is a wide range of the cytochrome P450 enzymes. They are extremely non-specific. They accept many substrates and they can hydroxylate very unreactive CH bonds. Okay. So, these are the classic examples of CH activation by nature. And you know, in organic chemistry, these days a lot of CH activation chemistry are coming up, and usually they use transition metals, and where from this idea has evolved. It has evolved from this cytochrome P450 chemistry. Okay. Here they use the iron. I have told you about how this iron ferryl oxo chemistry that is utilized to ferryl oxo can do lot of things. One is that oxidize the substrate and the oxygen comes out as water, that is called oxidases, and in some cases one oxygen atom is incorporated in the molecule and other oxygen atom goes as water, that is called monooxygenase, and then dioxygenase means both the oxygen atoms are incorporated into the molecule. Okay. So, that is dioxygenase. So, cytochrome P450 has different types of activities. One type of activity is they act as monooxygenase, because we need the hydroxylation. Okay. So, monooxygenase is basically an iron based enzyme. We do not want to go into the details again. it is a feryl oxo species like this. I think I may have the yes, the mechanism is shown here. This is actually different books have different ways of writing this. Let me see whether I can simplify the whole thing. The feryl oxo species, once it is formed, then you have this drug which is attached to a H. So, what will happen? The feryl oxo will go to OH, and so basically this is now, you can write this way, that one electron. So, it takes the hydrogen and the drug becomes the radical. So, the drug, whatever drug it is, it has become a radical, and which, and you have to be careful what is the oxidation state of iron, that is important. The oxidation state of iron, let us see one electron has gone here and iron gets back its electron. So, the oxidation state now is 4, and then this goes out and conjugates with the drug. So, the drug becomes hydroxylated, and the iron now again goes back to 3. The iron goes back to 3. Okay. So, that is the mechanism, but the mechanism, I think we have seen this mechanism time and again, one in penicillin biosynthesis and also in, I think, some other metalloenzyme, we have seen that how this feryl oxo species can carry out different types of reactions. Okay. Then now, what type of functionalities are involved in this hydroxylation business? RCH3, just aliphatic, that can be converted to RCH2H, and then there are enzymes which can take it up to up to the carboxylic acid, because carboxylic acid is more polar, more soluble in water. So, that can happen. Alcohol dehydrogenase is there, that converts it to aldehyde and then to the acid. And then you can have cyclic systems that can be hydroxylated. You can have aromatic systems, benzylic, so that can be hydroxylated. Ketones can be hydroxylated, alpha hydroxylation. Acetylenes, because of the propargylic hydrogen, that is the weakest bond here. So, that will be hydroxylated. So, these are different types of hydroxylation reactions that are carried out in the liver by the cytochrome P450. Okay. Now, there is another enzyme which also is basically, that also converts or degrades the molecule into certain products which are ultimately become water soluble. One is cytochrome P450, and here there you have seen that after the hydroxylation, in some cases they can be oxidized to the aldehyde followed by the acid by alcohol dehydrogenase enzymes. Okay. And acids are more soluble. Another class of enzyme which are called monoamine oxidase. Monoamine oxidase. Okay. What it does? It takes monoamine. Monoamine means primary amine, but they do not call it primary amine oxidase, they call monoamine oxidase. Okay. So, monoamine oxidase, what it does? It removes the nitrogen and forms the aldehyde. Suppose you take possibly you can remember this molecule OH and then CH2 NH2. This is what is called dopamine. Okay. There are many drugs which are based on dopamines, some structural variations here. Okay. And so, they have to be metabolized, they have to be taken care of, and so also the natural dopamine, because when dopamine comes from, if you remember again, tyrosine goes to the dopa, and then dopa goes to the dopamine by decarboxylation, and then dopamine goes to norepinephrine and then finally epinephrine. So, dopamine concentration you have to maintain at a particular level. I told you that that is very important for persons suffering from diseases, they always check dopamine, level of serotonin, all these neurotransmitters, acetylcholine, everything. So, dopamine, there is what happens, dopamine is forming from dopa, and usually it is converted into epinephrine. But there is an enzyme which is called monoamine oxidase. What it does? It oxidizes any primary amine RCH2NH2 that goes to RCHO plus ammonia. Now, this is an oxidation reaction, because this carbon was basically attached to one heteroatom, now double bonded O, that means one oxidation has taken place, and then this can be converted into RCO2H. If it is a drug, then that RCO2H becomes water soluble. However, this monoamine oxidase also becomes a target for developing drugs acting against neuro disorders, where the disorder is lack of the concentration or lowering of concentration of dopamine. Dopamine concentration can be lowered if it is converted to epinephrine, but epinephrine is another natural neurotransmitter. So, that is not a problem, but when it is degraded by monoamine oxidase, then dopamine becomes is basically removed from that circle or the cycle of events, that tyrosine goes to dopa, dopa goes to dopamine, then norepinephrine and then epinephrine. So, if dopamine is oxidized before it reaches epinephrine or norepinephrine, so what will happen? You have a shortage, you have a lack of concentration of dopamine as well as norepinephrine and then epinephrine. So, that is a problem. Monoamine oxidase, what happens? It takes out the dopamine from circulation and converts it into the aldehyde. Okay. So, that means this goes to the aldehyde. So, that is what is done by the monoamine oxidase. Let me be little bit quicker. So, monoamine oxidase becomes a target for drug development also. And the mechanism of monoamine oxidase, very quickly I will show. See, this is the monoamine RCH2NH2, and this is called MAO enzymes, monoamine oxidase. So, what it does? It converts, as I said, the amine goes to the ammonia and this is converted into aldehyde. Okay. This reaction requires oxygen, it is required, and water. Basically, what it does? It first goes into an iminium ion, and then water comes and hydrolyzes it to the aldehyde. Okay. That is the mechanism, and this is suggested to be a one electron type reaction. That means, first R CH2NH2 that goes to. So, if it releases one electron, see, you have to oxidize it, that means electrons have to now be released. The question is, there is a question, who takes up the electrons? Okay. Who takes up the electrons? It is, you know this, FAD. What is FAD? Flavin adenine dinucleotide. Okay. So, that takes up this electron and then it goes to RCH2 NH2 dot plus, and in the process FAD goes to FAD dot. So, one electron goes from the nitrogen, because that is the where it is most available, and then what happens? FAD dot, then to cut the story short, that goes to, it loses the hydrogen. This FAD dot takes up the hydrogen from the alpha carbon, and then that becomes a dot. So, these two dots combine and make this one, the iminium salt. Okay. And in the process, ultimately FAD becomes FADH2. FAD becomes FADH2. So, because two hydrogens have to be removed, one from basically one from here, another from here, and so that is the FADH2, and this ultimately hydrolyzes into this. So, monoamine oxidase is a very good target, and you see some of the drugs that have been shown here. One of the drug is based on simple organic chemistry, tranyl cypromine. It is an antidepressant drug. What is the mechanism you can guess that once the nitrogen becomes nitrogen positive, a radical, and because there is a cyclopropane, I have told you that the radical can be established by putting a cyclopropane alpha to it. So, it will open up like a radical clock, and as it opens up, the radical goes to some other place, and that radical then attaches to the again to the flavine, but at a different position. So, the whole cofactor is attached to the to this molecule. That means, it is a kind of suicide inhibition, that the normal reaction takes place, the first electron is removed, but then as it is removed, it is like alpha, that cyclopropyl methyl radical. So, it is cyclopropyl amine radical, that also opens up immediately. Okay. So, there are many kinds of drugs. There are two types of MAO inhibitors are there, MAO A, MAO B. If you are interested, you can go through it. Just one point I want to tell is that these MAO enzymes, if you that means to increase the concentration of dopamine or even serotonin, because that also has a free amine, then you have to use MAO inhibitors. But the problem, there is a slight problem, that MAO inhibitors will basically stop the metabolism of any primary amine, will slow down the metabolism, will inhibit the metabolism. And there is one compound which is called tyramine. What is tyramine? Decarboxylated product of tyrosine. What is histamine? Decarboxylated product of histidine. Okay. So, tyramine, which is present in many food products, and one of them is called cheese. Cheese has the highest level of tyramine. Okay. So, what happened, many people suffering from neuro disorders, they are taking these MAO inhibitors. At the same time, in our country we eat less cheese, but in foreign countries cheese is a very predominant item, and then they were taking the cheese, and cheese had lot of tyramine. The problem is, if tyramine levels increase in the blood, that ultimately has a cascading effect leading to heart failure, and this is what is called cheese effect. Cheese effect, that means a person who is taking antidepressant drugs based on MAO inhibitors, they should avoid cheese. Okay. However, medicinal chemistry has improved quite a lot. Now, we have selective inhibition of MAO A and MAO B. So, that cheese effect is, because westerners will eat cheese, we Indians do not eat cheese that much. Okay. But anyway, there are other food items also, they might have lot of tyramine. So, you have to be, people have to be careful, and also the reason for or what is cheese effect, if somebody ask you, then you can tell that it is basically because of the use of MAO inhibitors as antidepressants. Okay. The second type of removal of or the metabolism of or making the molecules more polar, making the drugs more polar is, I said, conjugation. In this case, it will be bioconjugation, because you are basically attaching to a drug another biomolecule. What is that biomolecule? Very simple. The biomolecule is nothing but a glucose derivative, a glucose. So, if you have an alcohol, if your drug is a carboxylic acid or an alcohol or phenol, then what happens? It undergoes what is called glycosylation. Glycosylation, that means it becomes, earlier it was, see, it was the uridine diphosphate to make it a good leaving group, uridine diphosphate. So, this is UDFP glucuronate. See, remember, glucose has a CH2OH here. In this case, it is carboxy. That means, it will be more soluble in water. So, what happens now? If you have ROH, then this forms the glycoside. Okay. If it is carboxylic acid, it forms these are O glycosides. You can get N glycosides, C glycosides, O glycosides, all are possible. Okay. So, through glycosylation, you can remove, because glucose has lot of OHs, as well as this is not glucose, this is glucuronic acid. That means, the primary alcohol is oxidized to the carboxy, and this is the agent for glycosylation, and the enzyme will be called a glucuronyl transferase, okay, because it is transferring the glucuronic group into the drug. Okay. That is one way. The other way is you can convert the OH or NH into sulphate, and there are sulfotransferase enzymes. They actually form, suppose ROH, that will be RO sulphate, and then R2NN sulphate, that means sulfonic acid derivative. Okay. And the reagent for doing the sulfur transfer is this compound 3' phospho 5' phosphosulphate, 5' phosphosulphate, and in the here it is adenine. So, the compound is 3' phosphoadenosine 5' phosphosulphate. Okay. And the enzyme will be called as sulfotransferase. A third one, the last one I will do, that is the glutathione conjugation. That means, if your drug is suppose an epoxide, then what will happen? An epoxide, then glutathione is a compound which is nothing but a tripeptide. See, this is what is glutathione. Glutathione is a L-glutamic acid linked to cysteine, linked to glycine. But remember, in glutathione it is not the carboxylic acid alpha carboxy is not which is attached by the peptide bond. It is the gamma carboxy that is attached to the cysteine, and cysteine attached to the glycine. This is a very good surveillance agent, because it is an antioxidant because of the sulfur, it can form the disulfide, and also it can form it can be a very good bioconjugating agent. Like if you have epoxide, then the sulfur being a nucleophile can open up the epoxide and form this, or if it is a alkyl halide, if the drug is an alkyl halide, then what will happen? Sulfur will undergo alkylation, but the whole thing is now soluble in water. Okay. Sometimes what happens? It can actually further hydrolyze by peptidases into only the cysteine with attached to the drug. Okay. And then what happens? This is acetylated. This is called mercapturic acid. Anyway, the important thing is that if you have a reactive functionality where glutathione can react, then glutathione, because of its it has got a sulfur SH, which is a powerful nucleophile. So, that attacks the drug and does alkylation or opening of epoxide, but ultimately there is a carbon sulfur bond formation, and then the glutathione part can be degraded to ultimately the cysteine, and that can be acylated and finally removed from the system. Okay. So, that is basically what is called metabolism. Excretion, ADME is absorption, distribution, metabolism. We have gone through metabolism, and excretion is usually through the kidney. Okay. So, that is the part, the PK part is done. Okay. It is mainly the PK part. You remove, see, we are not talking about pharmacodynamics, because dynamics has already been covered. Dynamics means what the drug does to the body. So, that means all the targets, and we have said anti-cancer, antibiotics, everything. So, that dynamic part has been taken care of, but this kinetic part now is told to you. Okay. In the next session, what we are going to do, that could be one of the penultimate session. We will discuss about something which is whether designing a drug again with all due regards to computational chemistry, everything, but ultimately there are certain other parameters which are very difficult to balance, like I said, lipophilicity, hydrophobicity, there are or hydrophilicity, they have to be balanced, but what is the value, how Lipinski arrived at those values, that it has to be log p should be less than 5, whether so those are basically it is an entirely different area of medicinal chemistry, that is called quantitative structure activity relationship. In the next lecture we will do that. Thank you.